Yes.
Yeah, yeah.
Okay, that's great. Hi everyone. Welcome to this new event of our Translational Neuroimaging Educational Program. In this program, we aim to progress the translational application of computational neuroimaging techniques to enhance human brain health. This education program is nested within the Enigma Lifespan Working Group, and it is a very inclusive program that welcomes every research at all levels of neuroimaging skills and from diverse training backgrounds, especially the early career researchers. The directors of these programs are Dr. Shalala Haas and myself, and we do have an international advisory committee. Please consider to join our mailing list to receive notifications about the updates. And we also have a YouTube channel, and so you can find the video recordings from our past events. So you don't have to record this event. We will do that for you. We also have a Twitter account. Please consider to follow us there. If you wish to share your works through this platform, and please get in touch with us, and we would be very delighted to host you as a guest. Today, I'm very delighted to introduce Dr. Junhao Wen from the University of Southern California. Dr. Wen is a new PI and a rising star of his research field. His research focuses on developing and applying artificial intelligence and machine learning techniques to analyze multi-organ, multi-omics, biomedical data for the study of human aging and disease. In this talk, he will present his recent findings on multi-organ biological age gaps, which were derived with machine learning techniques. He will discuss how multimodal biomedical and genetic data can be used to explore associations and causalities between human aging and disease. We also have an event scheduled at November 7th, and we will have Ilan from Amsterdam to introduce their recent work on polynomic scoring, which is a method designed to quantify disorder-related whole brain functional connectivity patterns. So please consider to register for it for free with this QR code. So I will pass the microphone to Dr. Wen. Dr. Wen, the microphone is yours now.
Okay, thank you. Thank you for the introduction, and I will share my screen. Can you see my screen?
Yes.
Great. Okay, thank you for the candid introduction. And I'm Junhao Wen, and colleagues and friends just call me Hao. And for today's talk, the title is Multi-Organ Biological Age Unlocks Causal Relationships Among Human Aging Diseases and Lifestyle Factors. And I'm currently as an assistant professor at the University of Southern California, and I'm the main PI of the Laboratory of AI and Biomedical Science. So for today's talk, I'm just going to introduce like the recent works from our groups, focusing on using imaging and genetic data to model human aging, which is a very common biomarker in the field, like brain age gap. And the key idea here is to try to leverage multi-organ and multi-omics data to understand human aging and disease better. So, like I mentioned earlier, neuroimaging-based brain age gap is very common and very hot, like a hot topic in the field. Normally, I will just briefly introduce this, what we call the brain age gap. So basically, we can train a machine learning model like traditional SVM, SVR, or like more cutting edge, like deep learning models, neural networks, using like brain MRI in neuroimaging data to predict the chronological age. And then we can define the brain age gap as the predict age minus the chronological age. A positive brain age gap means like your brain actually looks older than your chronological age. And on the opposite direction, if the brain age gap is a negative value, means your brain actually looks younger than your real chronological age. And brain age has been like a popular topic in the field, like I mentioned. And deep learning model, like neural network trained on like voxel-wise imaging, actually can obtain a very like pretty low mean absolute error, nearly like 3 years old on raw MRI data. And we, like previous research also found actually a trade-off between a low like MAE, meaning like your model fit like pretty well with your data versus generalizability across domains. That means like if we trained the model on a healthy control population, we derived the brain age gap. If we want to check whether those brain age gap differs between disease group across other domains, actually, the paper published 2 years ago from UPenn showed that if your neural network achieved much lower MAE, meaning like you may fit your data very well, but the generalizability of the model to like disease domain like Alzheimer's disease versus health control actually showed like lower separability between the two groups. Some other recent work also systematically estimate like benchmarked like brain age prediction tasks using different modality from MR modality like functional MRI, diffusion MRI, like DTI measurements, and also T1 weight MRI like cortical thickness, brain volume, etc. And with a set of like various like machine learning models. Another like more cutting edge like research area in the field is imaging genetics, which aims to try to combine like imaging data and genetic data to better understand like the molecular or biological pathway underpinning like that shape such accelerated or deaccelerated like brain age at population level. I think this imaging genetic research should be should be coming for like the first Nature paper published like in 2018 by Elliott and colleagues using like the UK Biobank genetic data. In this paper, they systematically linked like a multimodal, what they call like imaging derived phenotype (IDP) with whole genome sequencing data, like using imputed genotype data from the UK Biobank, and made the data publicly available. And after that, I think like brain age researchers also tried to perform like large scale GWAS, basically training the brain age gap at a phenotype and linking to common genetic variants, common SNPs. But most of the previous research endeavors focus on like T1 weighted MRI, like brain age gap derived from T1 with our brain volume, our cortical thickness as a feature. And those papers basically did not look at biologically or partially validate like those genetic signals, like the GWAS genomic loci identified in the large scale GWAS, because essentially as long as you have a larger sample, you can always find like significant signals. But to partially or biologically validate those signals is I think like the next step that we will, if we want to better understand like the molecular level or cellular level biological pathway of the aging. And also the previous studies, GWAS studies, did not focus on causal relationships, which may give like a very important clinical like implication with in the context of like multi-organ and the causality with lifestyle interactions. And of course, we know like that brain diseases like Alzheimer's disease is not just focusing on the brain, it also affects like some other organ systems. So previous studies did not look at this brain body like interaction, which has been a hot topic again like in the field, which I will discuss a little bit more like later. So the first paper I just want to briefly introduce is so inspired by this limitation in the previous literature, we systematically looked at like the multimodal brain age gap derived by using gray matter volume derived from T1 with MRI and DTI measurements like fractional anisotropy from the DTI diffusion MRI, and also the functional connectivity from resting state functional MRI. Sorry. So in this work, we first benchmarked the brain age prediction using like 4 different machine learning models and 3 different MRI modalities. The 4 machine learning models included 2 linear models, the linear SVR and the lasso, and 2 nonlinear networks, the vanilla multi-layer perceptrons and like the deep neural networks. But just to emphasize here, we use like the low dimensional features here based on like a brain atlas. We didn't like use the box-wise image, which could probably achieve like a better MAE. But as I mentioned earlier, a lower MAE does not specifically mean like higher prediction power across domains. And as we can see here, between the linear and the nonlinear model, actually, in our cases, a simple linear model obtained a like lower MAE and a higher Pearson correlation between the chronological age and the predict age. And of note, like between the two linear models, lasso seems like performing pretty good. So for all the subsequent analysis, we use like lasso as the main results. Then we also compute the Pearson correlation between the three multimodal BAG, green matter BAG, white matter BAG, and FCF BAG. I mean, as we expected, the green matter BAG and white matter BAG are highly positively correlated compared to other combinations. So after we derive those three multimodal brain BAG, we treated those three BAG as phenotypes in a genome-wide associations and identified in total 17 genomic loci. Those loci were largely, especially for the green matter BAG, were largely like consistent with those identified in previous studies. The most prominent or significant genomic loci that were consistently identified across different groups is a one on the chromosome 17, and on the figure below, we showed actually how those genomic loci, those SNPs within those genomic loci were previously linked to previous GWAS. Basically, this is the PHEWAS phenome-wide association lookup study in the GWAS catalog platform. And as we can see, on the left side, the 17th chromosome genomic loci, it was the SNPs inside this loci were linked to many different like clinical traits in the literature. For example, Alzheimer's disease was linked to this green matter BAG locus, and also many white matter BAG locus. And also some psychiatric disorder was uniquely linked to the green matter loci, for example, autism, schizophrenia, etc. And of course, clinical traits from other organs were also linked to the green matter and white matter BAG. For example, some traits relate to the kidney and relate to the lung and lifestyle factors. And uniquely, the functional BAG showed like a smaller significance compared to the other two brain BAGs, and it was previously linked to like sleep-related traits. And here again, we computed the genetic correlation between these three multimodal brain BAG, echoing like the previous phenotypic correlation. Similarly, we found very similar trends here. The green matter BAG and white matter BAG showed also the highest positive genetic correlation compared to other combinations. Another way to partially validate, like after doing the PHEWAS analysis, to validate like the genetic signal identified with the GWAS, is to find like potential drug repurposing implications. So in this gene drug disease network enrichment analysis, basically what we did is to take every gene linked to a specific brain BAG, like for example, the green matter BAG, MAPT gene, and we tested the enrichment of this specific gene in a gene site defined by the drug targets in the drug bank database, and we performed Fisher exact test, try to say whether this gene was overexpressed in this specific gene site of a specific drug. The example here, indicated by the right rectangle, is the RG6100. It's a molecular which was used to develop a anti-tau Alzheimer's disease drug, which targets the extracellular tau tangles, and was developed by like I think 2017. So here, basically, of course, we are not arguing to for drug repurposing for anti-aging drugs, which does not seem like plausible. It's just like implication at the gene level that those genes specifically linked to the multimodal BAG could have some implication for treating some other organ system diseases. And then we also perform the genetic correlation between the three multimodal brain age gap with some other clinical traits, including disease subtypes derived by AI, and also some cognitive scores like education, intelligence. And what we observe here is indicated by the star symbols that most of the three multimodal BAG are genetically correlated with the AI-derived subtypes. Like, for example, the gray matter BAG was positively linked to the AD1. It was basically an AI-derived subtype of Alzheimer's disease characterized by global atrophy patterns. And it was also linked to the white matter BAG, which makes sense because we know like gray matter, white matter disruptions were largely linked to Alzheimer's disease, and Alzheimer's disease and aging is like highly correlated. And another interesting finding is like the functional the FC BAG was negatively linked with education. I think the education trait here is the year that you finished your education, if I remember correctly. The second analysis, what we did is try to so we also estimate like the SNP-based heritability for the three multimodal brain BAGs, and they are all like significantly heritable. And here we are trying to understand the partition heritability enrichment, which means that whether the SNPs from a specific region, in total 53 regions without any specificity, that means it's not like cell specific or tissue specific. It's just like different SNPs along the whole genome. And we test like whether the region-specific SNPs are enriched compared to all the background SNPs. And as we can see, the green matter and white matter BAG were enriched at different regions regarding heritability enrichment. For example, the green matter BAG showed the highest enrichment level in the conserved region, which denotes all the SNPs that were conserved across different mammals along the whole genome. And I think that those SNPs are still like understood by the research community, and the exact function of this region has been largely unknown. And we also look like the partition heritability patterns at three different specific cell types. The first one is oligodendrocyte, the second one is astrocyte, and the last one is neuron. And as we can see here, let me move this here. As we can see here, the white matter BAG was enriched in the glia cell like oligodendrocytes, and the functional BAG was enriched in astrocytes. I think the result for the white matter BAG enrichment in the oligodendrocytes was consistent with a previous study led by published in Science by Dr. Bijin Zhao, which we performed the similar analysis in the DTI tracts. And also, I think the patterns between the white matter BAG and functional BAG enriched in the different types of glia cell, like oligodendrocytes and astrocytes, align very well with the architecture of the cell at the cell level, because we know like oligodendrocytes is over-enriched like in white matter, and the astrocytes may be enriched like in the cortical regions. Oh, sorry. Lastly, we also derived the polygenic risk scores using like the split sample GWAS setting. Basically, we split the sample into split 1, split 2 using split 1 as the discovery site and the split 2 GWAS summary static as the target. And as we can see, the overall prediction power, the incremental R square here shown here, is not that high and consistent with previous findings using the imaging derived phenotypes like the brain region, brain RI. And the green matter showed the highest incremental R square compared to white matter and FC, which are consistent with overall genetic signals identified in the GWAS. Finally, another way like to explore the genetic architecture of the 3-BAG, we performed 2-sample bidirectional Mendelian randomization between the 3 multimodal brain BAG and the several clinical traits, including disease endpoints and also lifestyle factors and some other biomarkers. As shown in the figure A here, the star symbol here showed there were significant results after multiple comparison. The other symbol denotes the nominal significance. As we can see here, the Alzheimer's disease is causally linked with white matter BAG. This is like reasonable because we know like, okay, Alzheimer's disease is a gray matter disease, but actually some previous research showed like macrostructural change disruptions in the brain actually could be an earlier marker than gray matter macroscopic atrophy patterns. And the other interesting finding is the triglyceride lipid ratio in VLDL is also causally linked with the gray matter BAG. Another significant finding is the gray matter, the breast cancer is causally linked with gray matter BAG. This is a little bit tricky to interpret this because we know like Alzheimer's disease, Mendelian randomization may be very sensitive to population bias or survival bias, and we know like cancer, this kind of clinical traits is very prone to this kind of survival bias. As I mentioned, like just now, like Mendelian randomization always overall has a very strong like statistical assumptions. So it's very essential to be cautious about like interpreting all those potential causal relationships. And normally a lot of like the good practice is to perform like a side of like sensitivity analysis to scrutinize the significant signals here. To demonstrate as an example, we showed different sides of sensitivity check for the causal relationship between the triglycerides lipid ratio in VLDL and the gray matter BAG. The figure B here showed the leave one side out Mendelian randomization, which means that each row we exclude these SNPs and re-evaluate the Mendelian randomization causal effects. As we can see here, there is not a single SNPs, our instrument variables here that overwhelmingly affect the causal relationships. And the figure C here showed another sensitivity check analysis where we tested like the single SNP based causal effects. As we can see here, there is a certain degree of like heterogeneity at the single instrument variable level regarding the MR effects. Some of the SNPs showed negative effects, but most of the instrument variable or the SNPs exert like positive causal relationship. And then to confirm this, we also performed a heritability test, and we found it was like the p-value for the heterogeneity of those SNP MR effects was nominal. I remember it was 0.04. So this could indicate some directional pleiotropy, which means that those SNPs serving as instrument variable may also link to some other confounders, where those confounder will subsequently affect those outcome in the case of the gray matter BAG. And figure D basically showed here the scatter plot. The x-axis here showed the SNP effect on the exposure, the triglycerides lipid ratio, And the y-axis here shows the SNP effect on the gray matter BAG. And as we can see here, basically the odds ratio of the Mendelian modulation, or the beta, is actually the slope of each MR estimator. Here we showed like the inverse variance weighted, which is the most common one used in the literature. And the other one is MR eager, which is another more like considerable or advanced MR estimator try to scrutinize the horizontal pleiotropy, which try to check whether the results violate the underlying MR assumptions. And from our analysis, we saw actually the intercept of the MR eager is a small positive value, but the intercept is not significant, which means the horizontal pleiotropy does not like bias the current results. And we also found a potential outlier for one of the SNPs, as denoted with the RS number here. And we rerun the MR analysis and still found the same like MR causal effects here. And finally, figure E here showed the Filner plot tried to assess the asymmetry of the SNP on both of the exposure variable and outcome variable, and we observed overall it's symmetrical, which indicates that there's no obvious violation of underlying assumptions. But we did find another outlier, and again we rerun the MR analysis by excluding this SNP, and the conclusion did not change. So basically, this is just those are just some practical ways to carefully scrutinize like your Mendelian randomization results. So so the follow-up question is like, we carefully look at like the brain age gap in the context of imaging genetics, try to understand the association and causality. And the next question we are trying to understand, okay, from our FIWAS, from our Mendelian randomization, we show that the brain BAG was not just causally linked to like brain diseases or other clinical traits with the brain, but also with other organ systems. So we are trying to understand, okay, can we generalize the finding from the brain to the other organs? And at that time, I think our collaborator from the University of Melbourne, Dr. Ye Alatian and Dr. Andrew Zaleski, they published this paper in Nature Medicine, derived the brain age gap from 9 different organs and linked them to lifestyle factor, clinical variables. But here they didn't like look at genetics, and we quickly like connect with each other and establish this collaboration to look at the genetic architecture of those non-biological age gap BAG of the non-human organ systems. So in these follow-up studies, first of all, we did like the genome-wide association studies and identified many new genomic loci that was previously undiscovered by previous GWAS. And of course, after deriving those GWAS signals, we want to validate those signals at multiple levels. The first thing we did is to perform again the FIWAS lookup analysis by looking at independent genetic signal in previous literature, previous GWAS. And here we take a more considerable approach. We considered fully the linkage, the disequilibrium, the LD, and we also performed the analysis across 2 different platforms. The first one is the GWAS catalog, and the second one is the GWAS atlas. The figure on the right side showed here basically the genomic loci, or independent signals for the non-BAG linked with previous literature, clinical traits in the literature. And the cloud plots just show the frequency of those traits in each organ category. As we can see here, we observed a very strong within organ specificity, which means the genomic loci linked to specific brain, a specific organ, are also linked to traits, previous traits in the GWAS were primarily of that organ. For example, we observed like a very abundant association between the metallic BAG versus traits of the metabolic system. Nearly more than 50% of traits identified in our analysis. So, but at the same time, we also, of course, observed like cross-organ talk, which means that, for example, the metabolic BAG are also previously linked to the hepatic system traits, immune system traits, etc. And then we try to derive several different key parameters of the genetic architecture of the non-BAGs. Here we estimate again the SNP-based heritability across the non-different organ systems. As we can see here, figure B, the brain showed the highest heritability estimate, followed by the eye. And among the body organ system, the pulmonary showed the highest SNP-based heritability. And of course, it's very tricky actually to compare the SNP-based heritability across different organs because the sample size here is not the same. So we also performed actually downsampled the body organ system to the same sample size of the brain because the brain basically has a smallest sample size and the transcript the same. To dig this deeper, we also look at like actually the absolute value of the beta coefficient. Basically, the GWAS is just a linear model, right? The phenotype is the BAG as a continuous variable and the SNPs as encoded by 0, 1, 2. So we look at absolute beta value, the magnitude of the beta coefficient, and we observed actually the same pattern. Basically, the brain BAG showed larger beta coefficient, followed by eye, then followed by other organ system. And we also look at like the effect allele frequencies, alternative allele frequency. In our case, we observe actually brain and eye showed higher for the significant SNP. Brain and eye showed higher alternative allele frequency. To interpret this, in our opinion, we think like the current sample size to may be sufficient to derive the high like high frequency based variants for the brain and eye, but maybe larger sample need to detect lower frequency, like rare variance signals for the brain and eye system. To confirm this, actually, we showed the trumpet plots for the 9 organ systems. The trumpet plot here showed the relationship between the effect allele frequency, the alternative allele frequency versus the beta value effect size. As we can see here, when the allele frequency goes smaller, more rare, the effect size become bigger, which is common in population genetics. Another way to validate those genetic signals across different organs is to look at the tissue-specific gene expression patterns. Here we look figure on the left side, the figure B here, we performed tissue-specific gene expression analysis using MAGMA and using the GTX54 tissues from the GTX project. As we can see, after correction for the multiple comparison, we found a very strong within organ specific enrichment patterns. That is, for example, the cardiovascular BAG on the most the dark green one, it was enriched. Their gene was enriched like in several different tissue related to the artery, like for example artery aorta, and for example the hepatic BAG denoted by the yellow color. The gene was enriched like in liver tissues, etc. And on the figure on the right side, we can also validate it as a SNP level. We know that we previously showed their SNP-based heritability was significant, and here we just look at like whether the tissue-specific tissue-specific SNP heritability analysis, whether those signals were enriched in a specific tissue compared to other background tissues. Again, we can see like very strong within organ specificity for these patterns. For example, again, the cardiovascular BAG showed like strong heritability enrichment in several heart or blood related tissue. A follow-up analysis was try to confirm the long-standing Chevreul conjecture, which basically propose or suggest the phenotypic correlation, the genetic correlation between two clinical traits largely mirror their phenotypic correlation. In this case, when some project we don't have the phenotypic relation information from at a population level, we can use the genetic correlation as a proxy to estimate the phenotypic correlation. To confirm this, we performed the pairwise phenotypic correlation between the non-BAG versus the genetic correlation between non-BAG. As the figure show here, we observe a very high concordance between the phenotypic correlation and the genetic correlation, and supporting the Chevreul conjecture in the literature. Finally, we also performed the genetic correlation between the non-organ BAG with other clinical endpoints, disease endpoints, and we found very strong, like within organ-specific like patterns. For example, the brain BAG denoted by the blue color was genetically correlated with many brain diseases like Alzheimer's disease and the subtypes, and little life depression, schizophrenia, and stroke. And another very interesting signal is diabetes. The diabetes are causally linked with many different organ systems. And of course, diabetes was primarily treated as a metabolic disorder, but actually it's also a systemic like conditions, which could explain why diabetes were associated with like many different organ systems like renal, pulmonary, brain, and cardiovascular, etc. Finally, we did the same analysis using the Mendelian randomization, but here we extend this causal analysis to the non-organ systems. I just want to highlight some of the signals here. The first one is, of course, Alzheimer's disease is very important and causally linked to multiple like organ system. As we expected, like we identified in our previous study, Alzheimer's disease is positively causally linked with the brain BAG, and brain BAG actually is also causally linked with sleep duration. And another strong signal is the body weight. So as we expected, the body weight is causally linked with many different like organ systems. For example, the musculoskeletal, the hepatic, and pulmonary BAG. So we also developed several different publicly available knowledge portals to disseminate our research finding and sharing data to the community. The medicine portal was designed specifically for the multi-organ research at the lab and with our collaborators. And the Mutate is a research portal to disseminate another paper looking at the causal relationship more broadly with other AI-derived endophenotypes in the group. And the Bridge portal was developed during my postdoc at UPenn to share the GWAS summary statistics of an AI-derived brain population. So to conclude, human aging and disease undergo like multifaceted etiologic mechanisms. And like in my personal opinion, nowadays we should go beyond the brain imaging only approach. Genetics can provide like new insights for actionable targets or interventions to ameliorate like overall human health in human aging and diseases. And we also maybe want to go beyond the brain. Within organ specificity and cross-organ talk need to be fully considered for precision medicine. And finally, I also believe AI shows potential in new phenotype discoveries to capture the neurobiological effects of disease and aging as individual level while modeling at multiple scales. The scales can refer to cross-organs or cross-omics data. So finally, I just want to thank you, my collaborator and mentor at the UPenn, Christos Davatsikos, and my collaborator from University of Melbourne, Andrew Zaleski and Yela Tian. And we are currently recruiting at all levels, PhD student and postdoc. So if you are interested in our research, just drop me an email.